afternoon session. It's my pleasure to introduce Maria Secrier, a good friend of mine, and she's a lecturer in computational cancer biology at the University College London. And she did her PhD at EMBL, European Molecular Biology Laboratories, and then later she did her postdoc at University of Cambridge, and also together with Cancer Research UK. And she also spent some time at AstraZeneca as a senior research scientist, so she has exposed to both academia and industry. And it's my pleasure, Maria, to give the keynote.
Thank you, Venkata, for this very kind introduction. I feel really honored to be giving this talk here today, and I want to thank the organizers for inviting me. Today, I want to tell you about some of the methods that have been recently developed to enable us to look at cancer development and progression, both from a genomic and from an environment point of view. So we've already heard a few talks today related to cancer, but I would like to take a step back and reflect on the ways we have been thinking about cancer development and how they have evolved over the years. So here you can see in this picture that actually cancer develops through a gradual accumulation of mutations that start to accumulate all the way from very early years in an individual through the individual's lifetime. And these mutations are caused either by intrinsic mutational processes or by different environmental and lifestyle exposures. And you do gradually accumulate these mutations up to the point where you have a mutator phenotype that will confer the cells a proliferative advantage, and this will lead to the development of a full-blown malignancy. Efforts like the Human Genome Project, as we've heard before in other keynotes, have been really instrumental, and despite their perhaps some of their disadvantages, they have also enabled us to look very carefully at cancer genomes, so to sequence cancer patients and understand how these mutations are driving cancer development. The first very large effort that tried to do this was from the US, the Cancer Genome Atlas, that aimed to sequence more than 11,000 people across 33 cancer types. And the main purpose of this effort, that was looking at whole exome sequencing, was to identify driver events in cancer. So by looking at mutations, copy number alterations, or fusions even, we wanted to identify these driver events, and they were really quite successful in doing so. So we now know a lot more about the different driver genes that are leading to cancer development in different tissues, and there is a long list of less frequent gene drivers. These efforts have also enabled us to understand that, first of all, there is a lot of interpatient variation. So for the same type of cancer, you have a lot of variation in the tumors of different individuals, but also through multi-region sequencing, we have also understood that there is a lot of intra-tumor variation as well. So if you look at different parts of the same tumor, you will see there are different subpopulations that are harboring different mutational landscapes. And these subpopulations are termed subclones, and they have enabled us to understand cancer from an evolutionary point of view. So these subclones are competing against each other, and of course the fitter ones will take over in the tumor and will lead to cancer progression. This has opened up a whole new area to look at cancer evolution through phylogenetic methods. So we can build phylogenetic trees that are tracing the different driver events and mapping them on the different branches of the tree to understand cancer and its development. And along with this also came the realization that there are different factors that are causing cancer, and these impose different mutational landscapes in the genome, and this is also relevant when we think about evolution. Now, another aspect of this intratumor variation looks at not only the tumor itself, but at the idea that the tumor is actually not an isolated population. It exists within its microenvironment, and there are all sorts of other types of cells, like immune cells or stromal cells, that have either anti or pro-tumorigenic effects. So it is important to take all this into account. And actually, there is a connection between the two, so they are not completely disparate factors in cancer development. And this has been made quite clear in just a few years ago when people started to look more into non-synonymous mutations of cancer. And what has become quite clear is that those non-synonymous mutations are actually causing aberrant peptides to accumulate in the cancer cells. And these aberrant peptides, since they are non-functional, they will be presented to the cell surface. And they will be recognized by cytotoxic T cells. So in a normal, healthy organism, these T cells would kill the cancer cells. But of course, the cancer cells come up with other mechanisms to avoid immune response. And this is by expressing some molecules called immune checkpoints. So this is the way they manage to evade immune response. Now, we can target these molecules, we can inhibit them, and this can be actually quite a successful treatment for certain subgroups of patients. And it turns out that the most successful subgroups are the ones where patients have a significantly high number of mutations. So this plot here is showing you the range of mutations, so the total mutational burden in different cancer tissues. And the cancers I'm highlighting are the highly mutated ones, which would also generate a lot of these aberrant peptides called neoantigens. And it has been shown through some really hallmark papers that these cancers really show very good responses to immunotherapy, so to checkpoint blockade. Now, of course, this is highlighting the fact that the way a tumor develops, the way it accumulates mutations, is important in the context of the microenvironment and in the context of the treatments that we can use for patients. So now I want to look a bit again at risk factors that I briefly mentioned before, and how they enable us to understand cancer development. When we sequence a human genome, what we get is a final mutational portrait of that cancer. So we see all the base substitutions and other types of variants that have accumulated in that genome over the whole period since the cancer commenced. However, these mutations are the result of different mutational processes that have acted upon the cancer cells from the birth of the cell. So they can be different processes. Some are, for instance, related to aging, like cytosine deamination. They can act for a longer period or for a shorter period but more intense, like smoking, or they can be intermittent, like acid reflux from the stomach. And now we have the computational methodology to actually look into these different patterns and the associated risk factors that generated the disease. So it turns out that the different risk factors that cause cancer generate certain mutational footprints that are quite specific. So you can think of these as footprints in the sand. If you see a human or dog footprint, it's very easy to recognize and distinguish them, even though you don't see the individual there anymore. So this is very similar. Different risk factors impose different mutational footprints in the genome, and you can use these footprints in order to understand cancer development and the different pathways that cancer can develop. So to give you an example, gastric acid reflux imposes this particular pattern of base substitution. So there usually we look at single base substitutions like T to G here, and they happen in a very specific mutational context. The adjacent bases are very important. We have this T to G substitution only in the context where the adjacent bases are C and T. And this, we believe, is associated to acid reflux. And that aging, for instance, would impose a really quite different mutational pattern. There are different ways we can look at this. One of the methods proposed a few years back by Mike Stratton's group in the Sanger Institute looked at decomposing the total number of mutations in a patient's genome into the different mutational processes that have specific patterns, and we call these mutational signatures. And we can look at what the contribution of each of these signatures is in the patient's genome, also taking into account a certain amount of error. The most common way to do this is through non-negative matrix factorization, where we are decomposing this matrix of total base substitution counts, so trinucleotide counts in the genome, into a matrix of probabilities of having certain mutations associated to different risk factors, and the exposures, the corresponding exposures of the signatures. And the idea is to minimize the difference between the observed mutational counts and the ones reconstituted by multiplying P and E. And this was derived, actually, this method was derived from the field of facial recognition. So it uses a probabilistic hidden variables model to identify the different features that compose a face and thus recognize the face in an image. So if we apply this kind of methodology, what we do is we take a cohort of patients, we apply this methodology, and then we can infer the different mutational signatures that are prevalent in that specific cancer type. And we can also see how prevalent they are in the different individuals. And to give you just a few examples of how this actually looks like, so the way we represent these signatures is through probability graphs. So each one of these bars is showing you the probability of having a specific mutation, C2T in this case, in the specific context. It's too small to see here, but these are the different nucleotide contexts that you can have. So it's a total of 96 in the genome. These patterns are really quite specific, so people have documented these, a lot of these patterns, and they are available in databases. So we have this pattern associated to aging or DNA repair and so on. So these mutational signatures can inform us about the way the cancer developed, but are they useful beyond that? So it turns out that they are starting to be, and the best example so far comes from Serena Nick-Zinal's group at the University of Cambridge, who looked at breast cancers and focused on women that have a BRCA1 or BRCA2 loss associated to breast cancer. So these women can actually benefit from a class of inhibitors called PARP inhibitors that are aiming to target the homologous recombination pathway, which BRCA1 and 2 are usually involved in. So by targeting PARP in that pathway, you can sensitize the tumor cells to the treatment, and this is quite effective. However, what Serena reasoned was that actually maybe we are missing some of the women that have defects in the homologous recombination pathways, but not necessarily in BRCA1 and 2. So those women could also benefit from PARP inhibitors, but we wouldn't detect them because we're only testing for BRCA1 and 2. So what she did was apply this mutational signature framework and search for the pattern of DNA damage that is associated with homologous recombination. And what they found was that in a cohort of 560 breast cancer samples, the gray ones are different scores assigned to patients that correspond to women that don't have a mutation in BRCA1 or 2, but could benefit from PARP inhibitors. So all the colored ones have mutations in BRCA1 and 2, but you can see it's a sizable slice of this graph that she identified as patients that could benefit from this type of treatment. And this is currently being tested in the clinic, and the results really look quite positive. Now, I want to tell you about a different story, one in esophageal adenocarcinoma that I have led during the time of my postdoc with Simon Tavare and Rebecca Fitzgerald. And this analysis was part of the International Cancer Genome Consortium, so another large consortium that aimed to perform whole genome sequencing this time of different cancers. So we had quite a large cohort of esophageal adenocarcinoma patients for which we performed whole genome sequencing. Now, esophageal adenocarcinoma develops from the normal epithelium. It's one of the two main types of esophageal cancer, and it's usually preceded by a precancerous stage called Barrett's esophagus that is most often caused by acid reflux from the stomach. And then it goes on to, some of these patients go on to develop cancer through different pathways, through P53 mutations, genomic instability, and so on. Now, for these patients, esophageal cancer patients, both adenocarcinoma and squamous cell carcinoma, the treatment options in the clinic are quite limited. So you can see that all of them can benefit from a combination of surgery, chemo, and radiotherapy. And really, the prognosis for these patients is quite poor. So in adenocarcinoma, only 15% of the patients go on to live for a period of 5 years. So we're clearly not doing the best job we could in terms of tailoring treatments for these patients. So our questions were if we could use this mutational signature approach to understand the risk factors of the disease, how it develops, and whether we could use these genomic histories to better reason about treatments in the clinic for these patients. So we took a cohort of 120 patients, we performed whole genome sequencing, and then we applied the mutational signature framework as explained before. And what we find is that there are 6 main mutational signatures characteristic for this type of cancer. The first 2 kind of relate to the same process. It's T2G substitutions in a very specific context, and we believe this is related to acid reflux. Then we also see signatures related to BRCA deregulation or other enzyme deregulation like ApoBec, an aging signature, and one signature that is not very well documented, so we don't really know the risk factor for that signature yet. And the idea was to look at the prevalence of these signatures in the patient's genomes and identify the dominant pathway through which the cancer developed. So if we cluster the patients according to the prevalence of these signatures, this is the heat map that we get. The rows are the different signatures, the columns are the different patients. The stronger the red color, the more prevalent, so the more mutations that signature is contributing to the patient's genomes. And you can really see that there are some clusters forming here. It's 3 main clusters that we then validated in TCGA. And we see the same patterns, really. One cluster is related to aging, so it has a prevalent aging signature. Another one is related to BRCA deregulation, so homologous recombination likely. And another one is the one with a dominant S17 signature that we believe is related to acid reflux. So we went on to describe these subgroups a bit further. And what we see in the first one that is related to the aging phenotype, we see a co-amplification of this class of proteins that are called receptor tyrosine kinases. And these are some of the proteins that are really leading to cellular proliferation, so they have an oncogenic effect. And they are often targeted in the clinic with inhibitors, but they're not very successful in esophageal cancer. So far, this lack of success of these inhibitors was not really explained in this cancer, but we believe that this co-amplification kinases may explain this. So in this heat map, you can see that these are the kinases and these are the downstream genes, and quite often you see co-amplifications in red color. The idea is that if you inhibit one of these kinases, there is still the other kinase that is amplified and is able to regulate the same pathway. The second subgroup, we see a lot of defects related to BRCA deregulation, but not necessarily in patients that have a BRCA mutation. So we actually, what we did was to look at all the pathways related to DNA damage repair, and we looked at the prevalence of mutations in these pathways. And in this plot, you can see the percentage of patients in blue indicated for each pathway that have defects in that particular pathway. So you can see P53 comes up, which is expected because it's the main driver in this cancer, but also the homologous recombination pathway, HR, comes up here. So we have the same situation as the one described for the method HR detect, in that these patients don't have BRCA mutations, but they do have defects in homologous recombination. So we believe PARP inhibitors could be effective for this subgroup. And finally, the mutagenic subgroup with a prevalent S17 pattern, which we believe is associated to acid reflux, and the significantly higher mutational burden. We also detected neoantigens. We predicted neoantigens through computational methods, and we see a significantly higher neoantigen burden as well. So this goes along the immunotherapy hypothesis, and we believe these patients could benefit from this type of treatment. So to summarize this first part, using this mutational signature analysis in esophageal adenocarcinoma, we find 3 subtypes of this cancer that have quite distinct etiology, and we believe we can better tailor treatment in this way for these patients. Now, we did validate these hypotheses using 2D and 3D cell models, but before taking this all the way to the clinic, taking this classification to the clinic, we really do need to think about certain things like cost reduction, because of course, if we're going to use this approach, whole genome sequencing or whole exome sequencing is still quite expensive. So we're currently working on shallow sequencing methods and random sampling of the genome to reduce the costs in order to be able to assess mutational signatures in a more effective manner. Now, this approach does confer an advantage over classical gene panels, and that is the fact that it can bypass spatial heterogeneity. So if in a gene panel you would search for a mutation in a certain gene, some part of the tumor may contain that mutation and some may not. But what we find with mutational signatures is that different parts of the same tumor in the patient have the same mutational signature profile, very similar mutational signature profiles. So we are spatially a bit more unbiased with this technique. Now, what about temporally? I just want to show you very quickly some of the findings we recently obtained from a collaborative study with the Sanger Institute and the Oxford Big Data Institute, where we sampled cells from patients' tumors, and we grew organoids from them. So these are 3D cell models of the tumor. We grow them over different passages, and we also sample them at different time points and perform whole genome sequencing of these time points of the organoids, as well as of the parent tumor. So then we can assess that really the organoids are really recapitulating the parent tumor quite effectively, and we can reconstruct clonal events in cancer evolution within a very restricted time window. We also use this to test drugs, but I don't have time to discuss this right now. What I do want to show you is the fact that there is quite an interesting clonal dynamics in this system. So this is the parent tumor, and these are organoids sampled at 3 different time points that are derived from this parent tumor. And the different pieces here are different subclones. So you have a blue, yellow, orange, and gray subclone. And you can really see that the proportions of the subclones are changing over time. So there is a dynamics of the subclones. And sometimes it's related to certain driver events. So the yellow subclone is increasing because it's driven by accumulation of new gene drivers, but some just oscillate randomly. So there is an intrinsic dynamic of the subclones, but what's very interesting is that the mutational signatures, however, are really quite stable. So we really see a very stable profile of mutational signatures over the time course. So this is suggesting to us that once mutational signatures are established in these cancer samples, then they tend to be stable for longer time frames. So we have within some limitations, a more stable system that is spatially as well as temporally. And this could suggest that it could be quite effective as a classification method in the clinic. Now, coming back to the mutagenic subgroup that had a high mutational burden and for which we believed immunotherapy could be effective, we really wanted to look further at this tumor immunity profile. So we do see an inflamed phenotype, but can we really characterize the spectrum of tumor immunity in this cancer? And how does it impact cancer progression in different mutational backgrounds? So what we know right now is that tumor immunity is built on a spectrum. Different patients have different types of immunity. You can have a very cytotoxic microenvironment or really a microenvironment devoid of any immune cells or something in between. And we are starting to understand this more and more due to different advances in both computational as well as experimental methodology to dissect the tumor microenvironment. So there have been different techniques that have been used both in bulk tumor data as well as single cell sequencing that have enabled us to gain a lot of resolution into the different cell types that populate the microenvironment of the tumor cells. We've also been able to identify new dendritic cell subtypes, for instance, or different phenotypes of the same cells, such as tumor-associated macrophages. Now, computationally, this has been primarily done in bulk tumors, or the idea started. Now single-cell sequencing is becoming very popular, but initially, the idea was that in a bulk tissue, you're always sampling the tumor. You're aiming to measure different parameters of the tumor. But by sampling a bulk tissue, you're always also sampling part of the microenvironment. And you can use the signals from the microenvironment to actually get a quantification of the cellular composition in this microenvironment. And that is because different gene markers are specific for different immune cells, and they would only normally just be expressed in these cells. So the way this is done in practice is usually through gene expression data. So you take, you have certain expression profiles specific for different immune or stromal cell types, and you either use methods like gene set enrichment analysis or other deconvolution methods to detect the fractions, to predict the fractions of different immune cells in the microenvironment. You can also do this with DNA methylation. It's not been as successful so far. Or you can directly stain the tissue with different markers, and that is quite effective, and some people try to use a combination of these methods. But primarily, most people currently use expression data. And really, the most popular method is some sort of deconvolution, where you have mixed gene expression data from a mixture of cells, but then you also know what the expression of different cell types should be, and then you try to assess the cell fractions of these cell populations. The most popular method to date is CyberSort, that came with the idea of using a support vector regression kernel to deconvolute these cell populations, and the data they used as training was just isolated distinct immune cell populations. Now, this has led to the different developments in the field, so there are more and more methods that are trying to do this, and some of them are actually combining a lot of publicly available databases that have expression data for these distinct immune cell types in order to define more robust gene signatures, so gene markers for different cell populations, and thus improve the predictions. Now, we do something similar in esophageal cancer, but not exactly, we don't apply any of those methods, because our aim is to assess the activity, so the functional states of the immune cells rather than just the abundances. And we also have some immune cells that may be quite specific to esophageal cancer, and they wouldn't be captured by any of those other methods. So we do use bulk RNA-seq data and a list of manually curated markers from the literature, and then use some scoring method to assess immune and stromal activity. The method we use is gene set variation analysis, and just very briefly, we rank order the genes according to their expression. Then we use a Kolmogorov-Smirnov-like statistic to assign some score distributions. For every cell category, you would have a score that deviates from 0, either on the negative or positive side, and that would be kind of deviations from the expected basal expression. So if we apply this method to esophageal cancer samples from the Cancer Genome Atlas, and we cluster the patient according to their immune profiles, we see there are 3 different types of immunity that arise in this cancer. We have a subgroup of patients that have a very inflamed phenotype, subgroup with non-inflamed, and something in between. Interestingly, but probably not surprisingly, for the inflamed phenotype, we see a lot of cytotoxic T cell activity, but it's always exhausted. So what this means is that the cells are always expressing those immune checkpoints that I was talking about, and trying to evade the immune system. And indeed, these checkpoints, you can see they're always higher expressed in this subgroup. Now, interestingly, we also see other intriguing dynamics of other cell subpopulations, and these are stromal cells, cancer-associated fibroblasts. And in collaboration with the University of Southampton, we did single-cell RNA-seq of esophageal samples, and we find 2 cancer-associated fibroblast populations, one with an extracellular matrix support role, and the other one are myofibroblastic calves. And what we see is that if we look at the progression of this cancer, cancers in more advanced stage tend to have a dominance of the CAF2 phenotype. So CAF2 seems to, there seems to be a switch towards CAF2 phenotypes in more advanced cancers. So we believe this is leading to cancer progression to some extent. So we were interested in this aspect of cancer progression, and we wanted to assess this a bit more formally. And because we mostly have primary tumor data rather than metastasis, what we can look at is at the local invasion phenotype. The way we do it is by developing a framework that looks at defining signatures of metastatic potential in these primary tumors. And these are based on genes that are highly active in metastasis. And we assign a metastatic score to every sample so we can classify it into low or high metastatic potential. And we did this, we applied it to different cancers. Here you can see in blue esophageal cancer. There is a sort of bimodal distribution that allows us to distinguish low and high metastatic potential samples. And this metastatic score seems to increase with cancer stage, which is what we expect. It is also prognostic of overall survival, and a higher metastatic potential is associated with higher myofibroblastic calf and M1 macrophage activity, and lower B cell and neutrophil activity. Now, how does this relate to the genetic background? For the sake of time, I'll just focus on the mutagenic subgroup, that was the one with acid reflux. And we see that these patients tend to have a higher metastatic score, so we believe maybe acid reflux is generating a more aggressive cancer phenotype. They also have a lot of inflammation in the microenvironment, which we would expect to some extent, but we also see a subgroup of patients that are really quite cold from this point of view. So the picture really seems to be much more complex than we originally anticipated. And what we suggest is that for proper targeting patients for immunotherapy, we really need to use more parameters. So on top of the signatures, we should use information about cytotoxicity, but also the signaling of the different immune checkpoints at the very least to identify the best patients that could be targeted. So to summarize the second part, we see that different mutational backgrounds actually generate divergent metastatic phenotypes, whereas the tumor microenvironment, as you've seen, generates these switches or increases in CAF2 populations and M1 macrophages that lead to convergent metastatic phenotypes. And finally, I would just want to highlight that while these approaches are helping us understand how to tailor therapy better to patients, we're still a long way away because so far I've focused in my talk and only that's all I will focus on is single base substitutions when we talk about mutational signatures, but large efforts from consortia have recently highlighted that there are different types of mutational signatures, also the ones generated by doublet-based substitutions, insertions and deletions, rearrangements and composite signatures. So the picture is much more complex, and this is generating much more diverse cancer developmental phenotypes. Even in esophageal cancer, we do have these three main signatures, but there are also some signatures that are less prevalent, and that's because the cohorts we've looked at are quite small so far. Really the potential to incorporate this will increase once we have more patients. Some of these signatures have been described by other groups, so they may have a greater role in decisions for therapy in the future. And now we're really understanding that we are shifting from the theory of clonal evolution that was proposed really quite a while back and was quite revolutionary by Peter Nowell, so the idea of different subclones that evolve in the tumor. And this is really shifting towards an understanding of the fact that different risk factors may shape the clonal composition of the primary tumor, and this is, along with the different components of the microenvironment in the tumor, may lead to different cancer progression phenotypes. So the different clones may migrate to different organs, and then they will also populate those organs in a different manner according to the microenvironmental niches there. So the picture is really much more complex, but we are moving with the computational experimental methods towards a more integrated view of this system. And this will hopefully help us better tailor therapies for patients in the future. And with this, I would like to acknowledge all the people that have been involved in this work, particularly Rebecca Fitzgerald, Simon Tavare, and Xiaodan Li, who has performed all the experiments in the mutational signature part and also the organoids story. And thank you for your attention.
Hello. Oh. Very nice talk, very interesting work. I have a question. Have you looked into, or is it known whether metastatic tumors share mutational signatures? From your last slide, I would guess it would be the case, but I don't know.
Yeah, that's an interesting question. It's actually not so much explored so far because the data sets with metastasis are quite limited. Okay. We have looked into this in esophageal cancer to some extent, and we actually see some dynamics. So we see some signatures are disappearing. So particularly the aging signature in esophageal cancer tends to disappear in metastasis. We don't really know why. So we're still trying to figure this out. But some signature seems to be more stable than others.
Thank you very much.
All right, thank you.